Myself, Mr. Mate H.T., Department of Botany, RBNB College, Sri Rampur. Today, we are going to study T.Y.B.Sc Botany, paper first, BO 331, Cryptogamic Botany. And from this paper, topic number 6, that is the study of a life cycle of the Puccinia gramineous triticale. Now, this life cycle is divided into the two phases, and these two phases are number one, that is the diplophase, and second, that is the haplophase. Now, in previous lecture, we had completed the stage number 1, phase number 1, that is the diplophase, and today we are going to study the second phase, that is the haplophase. Haplophase is nothing but the germination of a plus or minus strain mycelia, plus or minus strain basidiospore. They are haploid. So, basidiospores plus or minus strain basidiospore falls on the healthy barberry leaf. There they start to germinate. First they form the germ tip, and this germ tip directly penetrate through the upper epidermis of the barberry host leaf. And just below the upper epidermis, or in between the upper and lower epidermis, they form the haploid mycelium, and this mycelium may be plus or of the minus strain. So in this way, haplophase is get started by the germination of the haploid plus or minus strain basidiospore. And according to this, there is the two different types of the mycelia. Both are haploid, but some are of the plus strain and some are of the minus strain. Now we are going to next phase in the life cycle of the Puccinia gramineous triticale, that is the spermagonial or pycnidial stage. This is the upper epidermis of the barberry host, which is the dicot host. It is also called as the alternative host. These are the receptive hypha. Upper epidermis of the host upper epidermis. This is the fertile wall. These are periphyses. This is the receptive hypha. It is plus or minus. This is the spermatiophore. Here there is the ostiole. These are the pycnospore or spermatia. They are plus or minus. This is Pycnidia, or it is also called as a Pycnosaurus. Here there is the mesophyll tissue. This is receptive hypa. Plus or minus. Spermatia plus or minus. Very large number of the haploid basidiospores, that is from plus strain and minus strain, they germinate and develop the haploid plus or minus mycelium just below the Barbary upper lip, and just in between the upper and lower epidermis there develops very large amount of the haploid mycelium, and this mycelium may be plus or of the minus strain. Now, just below the upper epidermis, they form a special type of a structure, and that structure is called as the Pycnidia or Pycnosaurus. It is a flask shaped in outline. It has an opening toward the host upper epidermis. It is called as the ostiole. So, this is the ostiole, and it has a fertile wall. This fertile wall gives the three different types of the structures. From this, number one, just inside the ostiole, there are some filamentous structures are get formed. They are called as the periphyses. They are unbranched. They are filamentous. Now, from the lateral wall, there develops another filamentous structure. They are branched or unbranched, and they are called as the receptive hypa. So, these are the receptive hypa. They act as a female reproductive structure in the life cycle of the Puccinia graminis stratici. Now, this is the receptive hypa. It is plus or minus a strain. It is according to the basidiospore, which basidiospore is get germinated there. Now, the third structure which is developed by the fertile wall of this Pycnidia is the spermatiophore or the pycnophore. Now, this spermatiophore or pycnophore, they are elongated tube-like hypa, and at their apex they cuts up in chain small circular cells, and these circular cells are called as the spermatia or pycnospores. Again, they are of the plus or minus strain. In this way, very large number of these Pycnidia are developed in just below the upper epidermis in a number of leaves, in one lip or in many lips. Now, by chance, because of dispersal, the spermatization takes place. Now, here this pycnospore, they behaves as a male cell, and this receptive hypa behaves as a female reproductive structure, and after this spermatization takes place. So, this is about the spermatization. So, this is the receptive hypa. It represents the female reproductive structure. It is plus or minus strain. And this is the spermatia. It is also the plus or minus strain. And opposite strain, this pycnospore and spermatia come close in between their contact. Now the wall in between their contact is get dissolved, and the male content, that means the opposite strain nuclei with the cytoplasm, different types of the cell organelle enters in this receptive hypa. Now this male nucleus from the septal pore goes down toward the fertile wall of the pycnosaurus. That means here or here, and from here this nucleus, that is the male nucleus, is get migrated up to the lower side, and here the one stage phase of the pycnospore is get completed, which is called as the pycnosaurus. Now here each cell of this here, it is binucleated, and again in these two cells, one is of the plus strain and another nucleus is of the minus strain. Now we are going to the last stage, that is the aciospore stage. This is disjunctor cell. It is also called as intercalary cell. Finally, the male nucleus, which starts traveling from this receptive hypa, finally reaches up to the basal cell of the lower structure, which is called as the acidial cup or aciosaurus. Now it has its wall, and that wall is called as the peridium. So this is the peridium, and in the peridium there are the basal cell. In this way, all the cells of this basal cell becomes binucleated. That means in each cell there are the two nuclei, one is of the plus strain and another is of the minus strain. Now all this basal cell divides mitotically into another type of a cell, and that cell is called as the aciospore mother cell. Now this newly formed aciospore mother cell, it cuts up the two different types of the cells. The small cell is called as the disjunctor cell, or it is also called as the intercalary cell. It is very small and it is rectangular in shape. And there is another type of a spore. It is hexagonal, it is polygonal in shape, it is large, and it is called as the aciospore. Now this is the last phase, last spore stage in the life cycle of the Puccinia graminis triticii. Now it is a polygonal in shape, it has two nuclei. One is of the plus strain and another is of the minus strain. Now whole this is formed on the alternate host, that is on the Barberry lip. That means these ascospores are developed on the Barberry lip, that is in the Barberry lip. And from Barberry lip, these ascospores again infect the primary host, that is the monocot host, that is the wheat host, and again the dikaryophase starts. In this way, there are the two phases in the life cycle of the Puccina graminis triticale, that is the diplophase and this is the haplophase. But the diplophase starts here, that means at the basal stage condition, and because of this, the newly formed cell ascospores, they are binucleated.